Hi, greetings. My name is Ganesh Bala. I'm the manager of proteomics, metabolomics, and mass spectrometry facility at Montana State University Chemistry Department. We are located in the basement of the chemistry and biochemistry building. Room number 28 is where our mass spectrometry lab is located. We have over 12 different mass spectrometers. Each of them do different functions. Our major focus is proteomics and metabolomics. We also do quite a bit of small molecule mass spectrometry, reaction monitoring of enzymatic reactions, organic reactions, and several biological reactions using mass spectrometry. Hi, welcome back, and I'm going to talk more about the kinds of services that we offer through our facility. So first, talking about proteins, normally in a biochemistry or a molecular biology lab, people would run gels, SDS page or Western blots. We can actually cut the specific gel bands of interest, and we can digest them using specific enzymes and analyze them using mass spectrometry. We have state-of-the-art data analysis softwares that can piece those peptide fragments together and give you the proteins of interest or whatever proteins that we have detected. On the other end, looking at intact proteins, we have a state-of-the-art instrumentation. Here we have the Waters Synapse G2SI, which has an ion mobility capability. And what it can do for us is we can study the protein structures using this machine. We can fly the proteins in its intact state and study all the structure and dynamics of proteins, and whether it is in the form of a complex, a dimer, or a trimer, we can study with that machine. And this machine is equipped with a nanospray, so there is really picomolar sample amount is sufficient to do that kind of analysis. This is our sample prep area. So if clients who are not well versed with preparation of mass spec samples, we help them in preparing their samples. Also, students can come in and use this open access lab for preparing their mass spec samples. And this lab, we try to keep it clean to avoid contamination, so everybody can get best results from their sample preparation. And the samples are normally prepared in these vials, the HPLC vials. And we have a fume hood where if samples are corrosive or toxic in nature, people can use the fume hood. And we also have a hood which is a BSL-2 where if people are working with biologically hazardous materials, they can use that, which is situated in the first floor of the chemistry and biochemistry building. This is our main lab where we have most of the instrumentation hosted. And first we have here is our metabolomics workhorse. This is Agilent 6538 QTOF connected to 1290 Infinity binary pump, which is a UPLC scale instrument. So it can do high pressure, which in turn gives us high separation of a complex mixture. We have Bruker Maxis, which is our proteomics machine. So I showed you how we study the intact proteins using the Synapse. So in this machine, we study the proteins that we digest using trypsin or other enzymes, and then we do a long chromatography run and identify the peptides. Those will be pieced together to study what kind of proteins we have in our sample. This is our MALDI-TOF instrumentation, and this can analyze peptides, proteins of large molecular weight, over 100 kilodalton, and we can also study a lot of polymers, dendrimers using this instrumentation. This is our GCMS, Agilent 5975C, which is the mass spec component of it, which can do ETG and CI. And this is our GC system, which is the 7890A. It has two column capabilities, but we are using one column right now to study some of the lipid components that's present in serum samples right now. As you can see, there are some samples running. We use GCMS to study a lot of volatiles, non-volatile amino acids are converted to volatile silicon derivatives, and we have a NIST database that can help us identify the unknown compounds present in our mixture.